Bismillah ar-Rahman ar-Rahim. Today we are talking about oncology. We will have 3 lectures about this subject. And as we know that oncology is a science which deal with the study of neoplastic conditions. In these 3 lectures we try to know what is a tumor and how the process of carcinogenesis it will occur, and how the tumor it will develop, how it can spread, what are the methods which are used for diagnosis of such tumors, and how can we treat the tumor, and what do we mean by a tumor marker, and what do we mean by screening programs that are used to detect the malignant tumors at early stages to have an aim for cure in asymptomatic patients. A tumor is a swelling. It can occur anywhere in the body. It could be of inflammatory process, as we know that acute suppurative infection can cause infection and pus formation, which is encased or encysted within a certain area, and as this will occur, there will be formation of an abscess, and abscess will appear as a swelling, and as we said, that the swelling is a tumor. Or it could be to neoplastic conditions due to proliferation of cells at certain area which cause a growth, and the growth it can appear as a swelling, and the swelling is regarded as a tumor. Benign neoplasms is a condition which is diagnosed in surgical patients, and usually the benign neoplasm is composed of normally appearing cells that do not invade locally or metastasize to other sites. And as we know that the cells, it will look just like normal cell, and it is well encapsulated, as we will see, and it is very compact type of mass, and it will not invade the tissues or the cell. It will travel through a journey in the body to be implanted in somewhere else apart from the site of origin of that particular tumor. The cell will grow as a compact cell and remain there at their site of origin, and when these tumors are removed, the tumor usually it doesn't grow again back. In most cases, the tumor it will be cause its effect by pressure, and it cannot endanger life except in cases of benign brain tumors, because the brain is encysted within a very rigid case, which is the skull, and this will cause pressure on certain centers, causing neurological deficit, as affect the sensory or the motor areas, or it will affect the cardiovascular and respiratory centers in the brain, and it will become a life-threatening type of tumor. They are typically surrounded by an outer layer which is called the capsule, as in cases of lipoma, where it is encysted within a capsule, or it will stay within the epithelium, for example, in cases of uterine fibroid. The growth of benign tumors produce a mass effect, and it can compress the tissues and the surrounding tissues, causing some sort of pressure atrophy, and it can also impinge on a nerve and causing nerve damage, or it will reduce the blood supply by pressure over the vessels and causes necrosis and organ damage. The tumor of endocrine tissue, it might produce an overproduction of certain hormones, for example, in cases of thyroid adenoma, where it secretes thyroxine and to produce symptoms and signs of thyrotoxicosis, or adrenocortical adenoma or carcinoma of the adrenal cortex will produce cortisol and result in the formation of or development of Cushing syndrome. Many types of benign tumors have the potential to change into a malignant one. for example, lipoma in the interscapular area or in the retroperitoneum or gluteal region or intermuscular lipomas. They are more prone to change from benign lipoma into liposarcoma, and this is occurring through the process which is known as a tumor progression. The malignant tumors is composed of cells that exhibit uncontrolled proliferation, and it impairs the function of the normal organs by local invasion or by distant metastasis. These cells, they proliferate, and it has an uncontrolled proliferation, and it impairs the function of the normal cell by infiltration of the organ, nearby structure, or it will detach itself to circulate into or to reach another distant site from the site of its origin, and resulting in impairment of the function of the organ that it goes to. And these are called the distant metastasis. Most of the malignant cells eventually acquire the ability to metastasize, although some of them, they will remain at the site of their origin for a time, but in most of the cases they will metastasize later on. Thus, the major characteristic feature which differentiate benign from malignant tumors is that benign tumors, it is locally, it starts at a localized site, it has a capsule, it doesn't invade the tissue surrounding the tumor, and the cells, it will not disseminate or doesn't go far away from the site of its origin as the tumors of malignant tumors do. Tumor incidence, the most common tumors in our country, according to the Iraqi Cancer Registry between 2006 and 2009, that the most common tumor in our country is carcinoma of the breast. It constitutes about 16% of all tumors in Iraq. Lung and bronchus is the second most common tumor, and it constitutes about 8% of the tumors in our country, while leukemia is the third most common one, and it occurs in 7% of it regards constitutes 7% of the tumors, total tumors in our country. In male bronchogenic carcinoma, it is the leading tumor, and it constitutes about 13%, while urinary bladder leukemias and non-Hodgkin lymphomas constitute 37% of the tumor in male. In females, breast cancer, it constitutes about 1/3 of the tumor in our country, which is followed by leukemias, uterus and cervix, which constitute about 47% of the tumor collectively. How a tumor will develop, before we'll talk about the development of the tumor by process of carcinogenesis, we should know how the cell will develop. So we talk about cell development. The cell development passes into 3 steps during their development, which are under gene control. So the gene which control these processes, it will control the things, all the things. This includes the first step, which is cell proliferation. The cell will proliferate, and the cell will differentiate. Differentiation of the cell will occur by suppression of certain genes and expression of certain genes. So the cell, it will have its own biological activity and morphological features and biochemical activities as well. While these cells, once it will differentiate, it has a lifespan, and the lifespan of the cell is different at different tissues, and the cell will die at the end of their lifespan, and this is called a programmed cell death or apoptosis, just like the shedding of the autumn leaves, where the trees will fall, the leaves will fall from the tree to be reappearing again at the springtime. How do neoplastic cells differ from a normal cell? The neoplastic cell differ from a normal cell by alteration in the growth control, and it could occur due to either alteration in the proliferation or alteration of the cell death or alteration in the growth factor or factors which regulate the growth and response of the cell to certain substances. Or there is alteration in the cellular interaction, and this it occur between cell and the nearby cell, so-called cell-to-cell interaction, or between the cell and the stromal tissues that it is in contained with. So cell-stromal interaction, it might be altered, and this will affect or differentiate the neoplastic cell from a normal cell. The process of development of cell from a normal one to carcinoma or to a malignant one is called carcinogenesis. And carcinogenesis, usually it will pass into three major steps. The tumors are formed by the process of carcinogenesis. There will be alteration in the cell, would change this normal cell into a malignant one. Carcinogenesis, it is a multi-stage process by which it involves a sequential genetic and epigenetic changes that occur in the cell DNA, where each cell it will pass to a step and produce more advanced tumor. It is often broken into three stages. Three stages: the initiation, promotion, and progression. And there are several mutations which occur at each stage. The initiation is the step one in the development of the tumor, and where the first genetic mutation will occur. Once the initiation and genetic mutation, then the second step is a promotion, and the other one is a promotion. And promotion means there is a clonal expansion due to repeated division of cells, and this clonal expansion of the transformed cell into a visible tumor, this is usually still the cell is a benign one. In the process of progression, which is the third step in the development of malignant tumors, it may take place where more genetic mutations are acquired in the subpopulation of the tumor cell, and in the process of progression, the benign tumor or the benign cells, it will change into a malignant tumor and malignant cells. What are the factors which control the growth of a tumor? This could be divided into two groups. The first one is genes. The second one is the growth factors. And these genes, it can be divided into oncogenes, which are the genes which promote the cell proliferation and the instrumental instrumental in the development of cancer. For example, HER2, the human epidermal growth factor receptor type 2. And there are tumors which suppress the cell cycle by slowing the cell cycle, or trigger apoptosis, the programmed cell death. And these are called tumor suppressor genes. So there are oncogene and a tumor suppressor gene, and each one, it will antagonize or the effect of the other. In cases of tumor suppressor gene, the best example is BRCA1 and BRCA2 genes, which are usually, if it undergo mutation and deletion, it can make such patient more prone to develop carcinoma of the breast. And the p53 gene, which is also a tumor suppressor gene, if it undergo deletion, it will also predispose the patient to develop malignant tumors. This tumor suppressor gene will help in control the response of the cell to the protein, which is called a growth factor. So it will alter and control the effect of a growth factor on the cell. When the growth factor comes in contact with the receptor, the cell is stimulated to begin division more rapidly. So these tumor suppressor genes, it will affect or control the response of the cell toward these factors. The factors, the growth factors and their receptors is another type of the another factor which control the growth. The growth factor can stimulate the cell to proliferate, and the modulation in the receptor in the cell, it can also alter the cell and make such cell to undergo malignant changes. So we have factors which affect or which can result in the production or proliferation of cells, which are called oncogenes, and it is called switch on factors. And we have a switch off factor by which the tumor, it can be suppressed, and these are the tumor suppressor gene, including P53 and BRCA1 and BRCA2 there, and there are a lot of examples of that. The growth control of the tumor could be done by number one, increase or decrease the growth factor receptors or alteration in the receptor itself, including the number as well as the sensitivity of the receptor. And the second one is the synthesis of a growth factor by a cell. Either it will affect the same cell, which is called autocrine effect, or it will be produced by one cell and affect the nearby cell, and we call it paracrine effect. Third one is by excess or modify growth control proteins, which are oncogenes. So these factors, it will affect the growth of the cell and proliferation, and it will also exhibit the process of oncogenesis. This is an example. By this slide, we can see in autocrine signaling that the cell, it will secrete substance that it will affect the cell itself. So it has an autocrine signaling. And the second one, when the cell, it will produce a substance that it affects the nearby cells, and this is what we call it paracrine signaling. And the other one, which is the endocrine signaling, where the cell, it will produce substance that it circulates in the blood directly, it goes to the blood directly and circulate to affect other organs, which is very far from the site of their secretion. For example, the ACTH, the adrenocorticotropic hormone secreted by the pituitary gland, which affect the adrenal cortex to produce cortisol, or the thyroid stimulating hormone, which affect the thyroid gland to produce thyroxine. The causes of development of malignant tumors and cancer, they are not well known, but there are a lot of etiological factors that it can predispose such patient to develop a tumor cell. One of the factors is genetic factor. So there are diseases which are inherited, and it can be related to genes, like familial polyposis coli, which occur in families where the patient develop hundreds of polyps, adenomatous polyp in the colon, and the incidence of change of this adenomatous polyp into malignancy is 1%. So if you have 100 polyp, you will have 100% change in the adenoma into a carcinoma. And breast cancer, it has been found that 5% of patients with breast cancer have genetic predisposition, and the patient usually have a family history of first-degree relative who have cancer. And then if you do BRCA1 and BRCA2 gene assessment, I will find that there is a mutation of these genes or deletion of these genes, which are tumor suppressor genes, which will, if it get deleted, then the breast cell, it change into a breast cancer cell. And the other one is the familial retinoblastoma. It is a well-known example of development of carcinoma in families, and it can be related to an inherited factor. Chemical substances has been implicated to produce cancer, like nitrosamine, which is present in tobacco leaves. It can predispose the patient to develop lung cancer. And patient who works in aniline dyes, they will have aromatic enzymes within these dyes, which make this patient more prone to develop bladder carcinoma. Ultraviolet light is a well-known factor that can result on exposure for a long period of time for a malignant melanoma and basal cell carcinoma of the skin. Ionizing radiation, it can predispose the patient to develop thyroid cancer. So if you have Hodgkin lymphoma which have been treated by ionizing radiation to the neck, the patient he is more prone to develop thyroid cancer than those patients who have no extent or no exposure to irradiation. And patients or people who work in the radiation departments, they will have higher incidence of development of skin cancer, leukemias, and lymphomas in such people. Viruses have been claimed to play some role in the development of malignant tumors, especially the nasopharyngeal tumor, and this occurs usually in patients with herpes zoster infections. Cellular instability can result in development of skin cancer, and a very well-known example of such a tumor is the margarine ulcer, where it has been claimed that the scar is a stable tissue and has been found that it is an active tissue that undergo several changes during the patient life. And this instability of the scar will make this scar more prone to change into a malignant squamous cell carcinoma at the site of the scar, and the patient will develop what do you call it, margarine ulcer. Osteosarcoma is also occurring in area of the bone which is the most unstable one, where the bone development it will occur. Thank you very much for listening.